I'm Andy Gao. I am a postdoc researcher at John Curtin School of Medical Research, Australian National University. My research has been focused on T and B cell collaboration in response to malaria infection, a parasitic disease that is transmitted by mosquito. Well, the first project is about a type of immune cells called a typical B cell, which has strong association with malaria. So a typical B cell has been found that express surface markers CMC and transcription factor TBET. However, little is known about what drives a typical B cell formation, and also we have limited knowledge about what's the function of typical B cells, especially during malaria. My study has shown that transcription factor ZEP2 can promote a typical B cell formations, and by knocking out ZEP2, we discovered that there's a completely abolishment of not only the ABC formation, but also there's an impaired TFH formation, and also there's impairment of the germ center B cell formation and the antibody response. So in summary, this study is one of the first to report that ZEP2 is the key transcription factor that can drive ABC formation, but also reveal the function of those ABCs, especially in infectious diseases like malaria, that its role is to promote the TFH formation and eventually leads to an optimal antibody response. Then for the second project, we asked the question, so how does a B cell can take up the antigen from a large complex pathogen and then present to T cells to induce a optimal antibody response? Because we know that this process, this T-dependent antibody response process, is largely or predominantly studied in viral infection or proteinization model, but little is known about this process in terms of large complex pathogen. So here, we first discovered that B cell, antigen-specific B cells, instead of taking up the whole malaria parasite, a sporozoid malaria parasite, instead it spread on its cellular membranes onto the surface of the sporozoids and use this kind of manner to take up the antigen. In further study, we've shown that during this process, the B cell not only take up the cognate surface antigen, but also it can take up the bystander surface antigen, but not the internal antigen from the parasite. In summary, here we discover that in terms of large complex pathogens, the antigen-specific B cells can have limited access to only the spatial linked antigens to attain T cell help. Such observation can also offer the hypothesis for clinical observation that the host sporozoid vaccines have limited efficacy in malaria endemic region, and we guess this is because the malaria exposure can actually induce those T cells that are specific for cytosolic antigens outweigh those for targeting the surface. So if you have those B cell targeting the surface antigens, they may have a very difficult time to find those proper T cell helps, and thus this will lead to an impaired antibody response. In summary, my research has been focused on T and B cell collaboration in response to malaria infection, and I'm really looking forward to discuss more science with you in the conference. Thank you.